Hi, Pratibha. Good evening. Good evening. Good evening. Good evening to all. Yes. 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 Yeah, I'm fine, Pratibha. I'm fine. How are you? Sandeep has also joined. Sandeep, good evening. Yes. So, am I visible? Okay. Okay, dear. Okay. So, am I visible and audible to you, all of you? And just let me know whether my screen is visible too. Yes. Yes. Okay, let's start the class today. So we were discussing about the biomolecules. Yeah, okay, okay. Thank you, thank you, thank you so much. Okay. So we were discussing about the biomolecules in the last class, right? So we have completed carbohydrates and we are in proteins. Just a second. So in proteins, we were discussing amino acids and their classifications, and we are about to discuss peptide bond and classification of protein. Okay. So in the last class, I have told you that all proteins are made up of amino acids and basically many of alpha amino acids. Okay. And also we have seen their classifications also, like based on their acidic and basic nature and on the basis of structures and all. Okay. So essential and non-essential amino acids we have seen, and acidic, basic and neutral amino acids we have seen. And today we are going to see the peptide bond and classification of protein under this main protein topic. Okay. So moving on to the peptide bond. So all proteins are the polymers of alpha amino acids. So they are connected to each other by a peptide linkage, or we are calling that linkage as peptide bond. Okay. And how we are getting that peptide bond? So let's take one amino acid, which is called as glycine here. I have to glycine. Okay. And glycine is treated with alanine. So alanine is the other amino acid. Now these two amino acids are going to react with each other, and water molecule is going to get eliminated. So on removal of H2O from both the amino acid, like we have to remove the OH from one amino acid and H from the other amino acid. So the removal of H2O leads to a molecule which is called as glycyl alanine. So in this glycyl alanine, you can see that there is a linkage between C double bond O and NH, which is called as peptide linkage. Okay. So the complete structure will be, it is C double bond O and NH. So this linkage, this system is called as peptide bond. Okay. So there is an amide. It is the actually amide linkage, right? C double bond O NH is actually called as amide, and that amide functional group is present in the protein chain now. So that's why it is called as peptide linkage. Okay. Or we can call it as peptide bond also. So is that clear? The formation of peptide bond from the two amino acids. So since this particular peptide bond is formed between two amino acid, it is called as dipeptide. So if we have two peptide bonds in a single molecule, then those two peptide bonds would be formed from three amino acids, right? So means it is called as tripeptide. Likewise, it goes on tetrapeptide and polypeptide chains we have. So is that clear to everyone? Shall I go for the next slide? So do you have any doubt? Yes, yes, okay. So here you see, so I took glycine, then I took alanine. Now the third amino acid, same glycine I took. Now I took 3 amino acids. From these 3 amino acids, 2 water molecules are going to get removed. The first is from glycine and alanine. The second is from alanine and the second glycine molecule. Okay. So what we get ultimately, we get glycyl, alanine and glycine amino acid. Okay. So 2 peptide linkages are formed from 3 amino acids. Okay. So this is called as tripeptide. So you see here. So what is a dipeptide? If an amino acid contains 2 peptide bond, then it is called as, if it contains 2 amino acids and there is only 1 peptide bond in it, it is called as dipeptide. Likewise, the 2 linkages are there from 3 different, the one which you are seeing here on the slide, it is a tripeptide. And tetrapeptide, pentapeptide, hexapeptide, it goes on, and more than 3 carbon or 4 carbon or 4 peptide linkages containing systems are called as polypeptides. Okay, clear? So coming to the classification of protein, so we are classifying this protein into 2. First is on the basis of their molecular shape. Okay. So on the basis of their molecular shape, it is again classified into 2. One is fibrous protein, the other one is globular protein. So we are going to see some informations about fibrous and globular proteins, and those informations are very, very important for you in this lesson from protein part. Okay. So let me go with the first point of fibrous protein. So in fibrous protein, you can see the polypeptide chains running parallel to each other. You see here. So this is one polypeptide chain, and this is the other polypeptide chain, which are made up of amino acids, and they are running parallel to each other. The structure doesn't seem like a parallel thing. It is not parallel in this way, but still it is parallel only. Okay. So they just slightly they bend. And in case of globular protein, the coiling occur in polypeptide chain. So you see, so here it is forming a spherical kind of shape. So the polypeptide chain is forming a spherical kind of, like we are getting this kind of structure. Okay, it is called as globular. It is forming a globe-like structure. And fibrous proteins are having fiber-like structure, and globular proteins are having spherical shape, the sphere shape, which I have already told now. And fibrous are insoluble in water and they are soluble. Globular proteins are soluble in water. Examples for fibrous proteins are keratin and myosin. So in your hair, wool and silk control, you can see keratin over there, which is present as a fiber protein. And in myosin, in case of muscle is also contains this myosin, which is a fiber-like structure protein. Okay. And coming to the examples of globular, insulin and albumin are the examples of globular protein. Okay. Insulin and albumins are the examples of globular protein. So all the 4 points, including the examples under this fibrous and globular, they are very, very important. Okay. So do you have any doubt here in this classification based on the molecular shape of the protein? If you're having any doubt, just let me know in the chat box. If it is clear to you, just type yes. I'll move on to the next part. It's clear, right? Okay. Fine. So on the basis of their structure, on the basis of molecular shape, we have divided them into 2, that is fibrous and globular. Now, based on their structure, it is classified into 4. Okay. So those 4 are primary structure, secondary structure. Secondary is again classified into 2. First is alpha helix. The other one is beta pleated sheet, and tertiary structure. And the last one is quaternary structure. Okay. So this is a classification based on the structure. Now we are going to discuss from primary structure till quaternary structure in a detailed manner with 1 or 2 points under each topic. Okay. So coming to the primary structure. So the primary structure of protein consists of amino acid linkages which are linked with each other in a linear specific sequence. Just you see this So all the amino acids, they are linked with each other in a linear fashion. They are linked with each other in a linear fashion. There is no branch. There is no branch over there. Okay. Then the sequence of amino acids creates a different protein. Suppose instead of having THR, in place of THR, if you replace by glycine, you will get some other protein. Okay. If you just replace all the amino acid linkages with some other thing, you will get different kind of protein. So you can create a sequence of amino acids, creates a different protein kind of structure. Okay. And coming to the secondary structure of protein, it consists of two parts again, right? The first one is alpha helix and the second one is beta pleated sheet. So alpha helix is a regular folding of backbone of all the polypeptide chains basically. So you can see here, you can see the chain, that polypeptide chain, and this polypeptide chain, they twist into a right-handed helix. Just you see this, it is twisted into a right-handed helix. Okay, it is a right-handed helix kind of structure, which is called as alpha helix. And the bonding which is present between the two foldings, that is the two, what can I say, the interaction which is present between this particular chain and this particular chain is a hydrogen bonding. So there is a hydrogen bond present between the two chains. Okay. So that hydrogen bonding is between the carbonyl group of one system, one amino acid, and the NH group of the other amino acid. Okay. So what is the name of the interaction present here? It is hydrogen bonding, which is very weak, but still that interaction only keeps the helix together. Got it? So this is about the alpha helix. So the right-handed helix, this point is very important. Okay, you have to concentrate on it and you have to concentrate on the interaction which is present inside this alpha helix. Okay. So shall I move on to the beta pleated sheet? I hope this alpha helix is clear to you. Shall I move on to beta pleated sheet? Yes. See the interaction which I have shown you. So this is the interaction. This is the C double bond O of one amino acid, and this is the hydrogen of the amino acid which is present here, and they are getting interacted with each other. Okay. Then coming to the beta pleated sheet, they are stretched out and laid side by side. They are forming a pleated sheets. You know, you all know what is pleated sheets, no? So you can just a folded sheet like structure it is having. So these are two sheets, two pleated sheets are here, and here also the interaction is intermolecular hydrogen bonding. So the sheets are holding together due to intermolecular hydrogen bonding. What does it mean by intermolecular hydrogen bonding? We have two different types of hydrogen bonding, no? One is intra and the other one is inter. So intra means the bonding, the hydrogen bonding within the same molecule. Intermolecular hydrogen bonding means the hydrogen bonding which is existing between two molecules. The hydrogen bonding which exists between two molecules. Yes. Yes, Sandeep. Good. The hydrogen bond which exists between two molecules or two different molecules. They are called as what? Inter. So here you see the hydrogen bonding is between two sheets. That's why it is called as intermolecular hydrogen bonding. Okay. So see, there is the these are the hydrogen bonding interactions which are shown here in terms of red lines. Yes, Ruchinita. There are two types. One is inter and intra. The other one is intra. Good. So between two sheets, there exists an hydrogen bonding called as intermolecular hydrogen bond. Okay. So in case of alpha helix, it is a right-handed helix, and hydrogen bonding is the interaction over there. Here, they are forming a sheet-like structure, which is called as beta pleated sheet, and the force existing between two sheets is called as intermolecular hydrogen bond. Okay. So coming to the tertiary structure, which is the third structure of protein. So this structure represents further folding of the secondary structure. So this represents the further folding. Yes, Sandeep? Yes, Sandeep, tell me. Yes, Sandeep, you have raised your hand. Just tell me. You have any doubt? The sheets are placed. What is your doubt? The sheets are placed side by side, or can you repeat? Yeah, you are asking about the placement of the sheets. Yeah, yeah. They are in the side by side only. So you can place like this also. You can place like this also. So this is one sheet, and this is another sheet. You can have left and right like this also. You can have left and right like this. So that will be the hydrogen bonding like this in case of left and right position. Or if you are keeping the sheet at the top and one more sheet is at the bottom, like you are seeing in this picture, then the hydrogen bonding will be in this direction. Okay, it can be of any direction. Left, right, up and down. It can be like this also in a sideways manner. Okay, direction is not the matter. Got it? It is not necessary or compulsory that you have to place the sheets only in the up and down manner. So I hope this is the doubt you were asking, right? Yes? Yes? Yes. Just a second. Yes. So I'll go for the next step. Okay. So coming to the tertiary structure. So what is your another doubt? Yes. Why it is called as beta? So it is called as, so beta is the second Greek letter, no? Alpha is the first Greek letter and beta is the second Greek letter. So first they have discovered that alpha helix. That helix they have discovered. So they have called or named that as alpha. And after that only they come to know there is also some interaction between the amino acid or the polypeptide linkages between sheets. Okay. So since they have discovered this sheet in secondly, they have named this as beta. Since this was discovered secondly, they have named this as beta pleated sheet. Okay. Clear? Yes. Okay. 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 So I'll go for the tertiary structure. So tertiary structure represents the further folding of the secondary structure. So what is actually primary? Primary means just the linear linkages. So what is secondary? In secondary, there is folding like this. And tertiary is the further folding of the secondary structure. This is secondary. And if you still fold this, it is called as tertiary. Okay. So you can see. So this is the folding. So this is the alpha helix. And if you still further fold that, you will get the tertiary structure. Got it? So this only gives two major molecular shape, which we were discussing in the previous slides. Fibrous and globular proteins. Okay, fibrous protein and globular protein are the two major molecular shape which arises out of combination of two secondary. Combination means adding, right? Combination means adding. We are not adding these two. We are not adding these two, but during this addition, there is also some interaction which is existing between two, right? So it is not called as addition. It is not exactly called as addition. It is not exactly called as addition. Yeah, they are placed parallel only, but you see, there are polypeptide chains running over here. So we are not just basically adding. So a new bond is not formed between the two alpha helices. If you still further fold it, you will get this structure. Got it? You're not adding, the chain extends. The chain extends and they get coiled with each other. Okay? Okay. So now the interaction which is existing between the tertiary structure of protein are 4. So those 4 interactions are making this molecule, making this tertiary structure stable. Okay. So the first interaction is called as hydrogen bond again, and the second interaction is disulfide linkages. Disulfide linkages means there will be disulfide, sulfur linkages will be existing between the two system. That is, they'll exist between here. From here and here, you can see some disulfide linkages. Got it? And third is the van der Waals interaction, which is the very weakest force of interaction. So this force is also existing here. And the fourth one is electrostatic force of interaction. So what is in the electrostatic force? It is called as ionic force. Electrostatic is otherwise called as ionic force. So since we have C double bond O group and also NH group in the amino acid part, so the oxygen is the negatively charged center since it is more electronegative. Okay. And the nitrogen is also an electron rich center. But when we have NH2 attached with the C double bond O system, this, just a second, I'll write like this for you. So let me take a glycine for you. So in glycine, what we have COOH attached with a carbon, and the carbon is attached with NH2, and the other 2 sites are occupied by 2 hydrogens. So this is glycine, right? So this part is the acidic part, and this part is the basic part. So what happens here? The lone pair which is present on the nitrogen will go and attack the hydrogen of OH, and the electrons which is present here on the bond will move towards the oxygen. So what you will get a zwitter ionic kind of structure, no? Which we have discussed in the previous class. So it is O minus, and here we get N plus. So we are having negative and positive centers. So this will act as an ionic force of interaction between the 2 amino acids. Got it? So there are 4 forces of attraction which is existing between the tertiary structure. So hydrogen bonding, disulfide, van der Waals, and electrostatic forces of attraction. Okay. So you have to study which type of interaction is present under which structure. Got it? Shall I move on to the next structure? Quaternary structure. Shall I move on to the quaternary structure? See, Sandeep, 4 different types of interactions are existing in tertiary. One is hydrogen bonding, the other one is disulfide, the third is van der Waals, and the fourth is ionic force of interaction. Ionic in the sense the carboxylic acid part is carrying the negative center and the NH3 is having the positive center. No. So those 2 ionic interactions are also present between this. Got it? Yes. Now coming to the quaternary structure of protein, it is composed of 2 or more polypeptide chain. 2 or more polypeptide chain. See, so the green color is on polypeptide chain, the other one, the orange color which you can see, that is the other polypeptide chain. So 2 or more than 2 polypeptide chain constitute this structure, quaternary structure. So it is a complex, very complex structure from which you cannot see that which link is going to the from this part to the other. You cannot find out the starting point and ending point of all the polypeptide chain. because it is a very complex structure. Okay. So I'll give you the 4 structure of protein here. This is primary, which is having the linear fashion chain, and this is secondary. This is your tertiary, and this is your quaternary. So in quaternary, you see this is the first polypeptide chain, and this is your second polypeptide chain. So 2 polypeptide chain constitute 2 quaternary structure here. So more than 2 also I can use. Okay. So this is a representation of all 4 types of proteins. So any doubts so far here in the structure of proteins? Okay. Okay. So the next topic under protein is denaturation of protein. So what does mean by denaturation? Losing its nature. The one which is losing its nature is called as denaturation, right? So how this protein is going to get denaturated? So it is due to the change in temperature or pH, protein loses its biological activity. Okay. So due to temperature, if you increase the temperature or if you increase or decrease the pH value, then the protein can loses its biological activity. I'll show you an example. So this is the protein. It is functioning now. So this functional protein has some biological activity. Yes, good. So if I change the temperature, okay, if I change the temperature or pH, what happens? The protein chain, the globules gets unfold. You see? So there are interactions present, no? There are some hydrogen bonding interaction present, disulfide linkages present, and those linkages are now getting break here. So what happens now? The globules get unfold. So because of this, what happens? The helix, the alpha helix, everything gets uncoiled. They get uncoiled, and because of this, the protein loses its biological activity. Okay. So this protein is called as denatured protein. This is called as denatured protein. Suppose you can take egg. So egg consists of proteins, right? So we are boiling the egg. So during boiling, what happens? The protein chains, they are getting a break, and the protein loses its biological activity, which is called as denatured protein. Okay. So the increase or decrease in temperature is called as physical change we are doing, and the increase or decrease in pH is called as chemical change. So a protein can lose its biological activity by 2 changes. One is physical change like change in temperature, or chemical change like change in pH. Okay. So any doubt in the denaturation of protein? No doubt. Shall I go with the summary of protein? Yes. So let me summarize the protein. So proteins are required for growth and maintenance. They are consist of alpha amino acid and peptide bond or peptide linkage connect each amino acid, and they form a polypeptide chain. And we have classified this into 4, and secondary consists of 2, alpha helix and beta pleated. So this is of protein, the second biomolecule. Okay. Now I'll go with the third, fourth, fifth, and sixth biomolecule. They are enzymes, vitamins, nucleic acids, and hormones. Okay, all are very, very small topics. So we'll just try to complete it. So we are going to see about these 4 enzymes, vitamins, nucleic acids, and hormones. Okay, so coming to the enzymes. So what are enzymes? Enzymes are biocatalysts. Enzymes are biocatalysts. This was the question asked in 2022 NEET. So they have given 4 statements about the enzymes, and they have asked you to find out the incorrect statement from the given options. Okay. So the first point is enzymes are biocatalyst. This was one of the options given there. And enzymes are responsible for smooth biological activities in the body. Okay. All the enzymes are globular proteins. You can see here. So they are having spherical structure, shape, and each enzyme is doing very specific work in its action. Okay. Each enzymes are very specific. Suppose if an enzymes are biocatalyst, why do we use the catalyst? To speed up the reaction, right? So each enzyme is very specific to a particular reaction. Suppose if we are doing oxidation and reduction, if you are doing a redox reaction, then you have to use the enzyme which can do that particular activity. Okay. So if all the enzymes cannot do all the reactions, only very specific enzymes can do very specific reactions. Okay. And let me show you the naming of enzymes. So first naming is based on compound or class of compounds upon which they work. Second is based on the reaction. So based on the class of compounds upon which they work, there is an example on the screen to you. So which is maltose. So this maltose on reaction with water, it forms 2 moles of glucose. So what is the name of the enzyme we use? Here we use maltase. So maltose, so the enzyme is acting on the reactant maltose. That's why it is called as maltase. So this is based on the class of compounds on which they are reacting. Okay. Now coming to the reaction part. So an enzyme, if it is doing both the oxidation as well as the reduction, it is called as oxidoreductase. Then that particular enzyme is called as oxidoreductase. Okay. This is based on the name of the reaction or the type of the reaction. Here it is doing redox reaction. So it is called as oxidoreductase. So here it is taking the name of the reactant upon which it is working. So it is called as maltase. Okay. Most enzymes names contain the suffix AS. So these are the very important and simple points under this. Clear? Shall I go with the next slide? Yes. So now coming to the mechanism of enzyme. So what is this enzyme doing actually? They increase the rate of the reaction. That is, it is increasing the speed of the reaction by lowering the activation energy. So let me show you a graph between reaction coordinate and energy. So this is the reactant energy and this is the product energy point. And now reactant has to get converted to product. Reactant has to get converted to product, right? For that, the reactant molecule has to get activated. Then only it can do the reaction and will be converted to product. So the energy required for the activation of reactant is called as activation energy. And for a particular reaction, a reactant is taking this much of activation energy, which you can see on the slide, without the use of enzyme, and it is getting converted to product. But with enzyme, if you are using some enzymes which are the biocatalyst, activation energy is reduced. So what happens? The reaction occurs very fast, right? The speed of the reaction occurs very fast. Suppose if a reactant is getting activated by taking this energy only after 1 hour for this particular reaction, if the energy is getting decreased or lowered, the particular reactant molecule can get activated within half an hour or 40 minutes, right? So what happens at the end? The rate of the reaction, the speed of the reaction increases with the presence of enzymes, which are acting as biocatalyst. Got it? So this is very important. Enzymes increase the rate of the reaction by decreasing or lowering the activation energy. Clear? Is the mechanism of enzyme catalysis clear? Yes. So the enzyme part is over. Very small topic. Now the fourth biomolecule of this lesson is vitamins. So what are vitamins? You can see a structure of vitamin here. Example of a structure of vitamin. They are the organic compounds required in the diet in very, very small amounts. So every day we need all kind of vitamins in our diet, which has to be taken only in very small amount. Okay. Excess of vitamins also leads to some disadvantages. So vitamins perform specific biological actions and They only maintain the growth and health of an organism. They perform biological functions. They help in maintaining the growth and health of an organism. Okay. Now I'll classify this vitamin into two. First classification is fat soluble vitamin based on their solubility, and the second is water soluble vitamin. So what are fat soluble vitamin? So where can you find those fat soluble? They are stored in liver and adipose tissue. I'll tell you, Sandeep. So fat soluble vitamins are stored in liver and adipose tissue. Examples are vitamin A, D, E, K. Very, very important. Okay. They'll give you 4 options and they will ask you to find out which among the following is the only fat soluble vitamin, or which among the following is not the fat soluble vitamin. Like that they will ask. So instead of getting confused with the fat soluble vitamin example and water soluble vitamin example, you just study properly the examples of at least any one of the classification. Okay. So under fat soluble falls on this ADEK. The remaining are water soluble only. So water soluble vitamins, they are readily excreted in urine and they cannot be stored in your body. Okay. So they are readily excreted. So fat soluble vitamins are stored in your body in liver and adipose tissue, whereas water soluble vitamins are excreted in your urine and it cannot be stored in your body. Okay. So it must be supplied regularly in the diet. Okay. So vitamin C, example. Yes, Sandeep. You cannot store vitamin C. You cannot store vitamin C. Okay. It does its action and it gets excreted. So vitamin C and vitamin B group. So they are water soluble. So where does this vitamin C present? In all the citrus fruits you are having, no? In lemon juice, orange juice, in amla, everywhere you have vitamin C. Those are except B12. Yes, exception is B12. So here you can take a note of it. So except B12, all under B group and C group, except A, D, E, K and B12, all are water soluble vitamins. Okay. So clear. Shall I go with some examples for fat soluble and water soluble vitamins to you? Classification is clear to you, right? So these points are very important. And here it cannot be stored. Examples are also very, very important. Got it? Yes. So coming to fat soluble vitamin, the first fat soluble vitamin given to you on the screen is vitamin A. Okay. So it is present in retinol, that is vitamin A1 actually. The sources of vitamin A are fish liver oil, carrots, and milk, butter. Okay, all these sources contains vitamin A. And if you are deficient in this vitamin A, you will get some diseases which are night blindness, xerophthalmia, etc. Okay. So mostly all type of eye diseases you will get if you are deficient with this vitamin A. Okay. So you have to eat carrot, you have to drink milk every day. You have to maintain your body. Okay. So I'll go for the next vitamin, which is vitamin D. It is cholecalciferol, in which it is present as vitamin D3. The sources of this particular system molecule is when you expose to sunlight in the morning, you can get provitamin D. You can get this provitamin D directly from sunlight. That too, the early morning sunlight gives you, okay, the vitamin D. So, and in fish also you can find this vitamin D. So all of you have to eat fish at least twice or thrice in it. Okay. And egg yolk. And egg yolk tanning is the side effect. No. I'm not asking you to stand in front of the sun for hours and hours. Okay. Just go and stand for 5 minutes. Just go and stand for 5 minutes. You will get it. Okay. And during our school assembly and all, we used to stand in the ground. At least That just stand for 5 to 10 minutes. That's enough. You'll not get that. Okay, fine. Now the deficiency of vitamin D leads to rickets, osteomalacia. Osteomalacia. You can see, right? It's a bone. So you can see the normal bone. You can see the osteomalacia, disease affected bone. So this type of disease you will get. Okay. Okay. I'll go for the next vitamin, which is vitamin E. It is alpha tocopherol. Is the compound which is present in vitamin E, and the sources of vitamin E are beet germ oil and sunflower oil. So deficiency of vitamin E leads to the red blood cell fragility and all the muscular weakness. So all the girls should be having this vitamin E. They should not go in deficient. Okay, boys also, you have to take care of this vitamin E part in your body. Otherwise, all your muscles will get weak and you can feel the muscular weakness. Okay, the students mostly don't get deficient with this vitamin. Yeah. Yes, Sandeep. Yes, Sandeep. Any doubt? Any doubt you have? What is your doubt? What is RBC fragility? So it is kind of RBC deficiency only. Fragility means all the red blood cells, what they can do, they can broken easily. Cells can broken easily. So RBC deficiency kind of. Okay. So the cells are not at a good condition. That is called as RBC fragility. Okay. Clear? Yes. And coming to vitamin K, which is phyloquinone. So all the green leafy vegetables contains vitamin K. So you all have to eat all types of greens in your home. Okay. It increases your blood clotting time. You see, if you are getting some injury, it increases your blood clotting time. If you are not good at having vitamin K, so you should take all types of green leafy vegetables at least twice or thrice in a week in your diet. Okay. Now coming to water soluble vitamin. So far we have discussed the fat soluble vitamin A, D, E, K we have discussed. Now coming to water soluble vitamins. The first one is vitamin B1. So thiamine is the system molecule and the sources are yeast, green leafy vegetable again milk and cereals, all type of cereals. Okay. These are the sources from which you can get vitamin B1. And if you are in deficient with this, you will get beriberi. You can see the beriberi disease on the screen now. You will get this type of disease if you are lacking with vitamin B1. Okay. Now coming to the next water soluble, which is vitamin B2, riboflavin. The sources are liver. Vitamin B2 is present in liver and it is in egg white, milk, kidney. It is present. Vitamin B2 is present in all these four, and the deficiency of this leads to chelosis and the burning sensation of your skin. So daily you all eat egg and drink milk. Okay. So it will green leafy vegetables. So these three are very important for your diet. Now I'll go for the next water soluble. Yeah, one more is also there. The digestion problem. Digestion problem you will get if you are lacking with vitamin B2. Shall I go with the next vitamin? Yes, vitamin B6 is there in pyridoxine. The sources are yeast, cereals, milk, and egg yolk again. The deficiency is convulsion. Convulsion means a fits kind of problem you will get. Okay, fits. So you see a person falls off, fits. And coming to the next one, vitamin B12, cyanocobalamin is the compound present there in vitamin B12. The sources are fish, meat, curd, and egg. Okay. So it comes under water soluble, but it can be stored. Vitamin B12 can be stored in your body. So fish, meat, curd, and egg are the sources. Deficiency leads to anemia problem, pernicious anemia. Okay, the person becomes anemic if he is in deficient with vitamin B12. Okay. So the next vitamin is C, ascorbic acid. It is present in citrus fruit, green leafy vegetable, and amla. The deficiency leads to scurvy, some bleeding gums. Okay, scurvy is a kind of bleeding gums. Clear? So any doubts so far in the vitamins? All the fat soluble and water soluble vitamins are clear to all of you, right? So Sandeep and the other person was joined from the ID Samsung. Is it clear? No doubt? Okay. You have a list of it in your NCERT book. So there will be one tabular column given in your book. So that's what I have discussed here with the pictures. Okay, for your better understanding, you can go through it. So you have to read each and everything which is given in that table. Okay. Examples and the compound which is present there in particular vitamin like ascorbic acid and vitamin C and the sources, everything is important. Since all the topics are very, very small topics, they can ask questions from any nook and corner of the book. Okay. Fine. I'll go for the next biomolecule, which is called as nucleic acid. So every generation of each and every species resembles its ancestors. You all may be well aware of this. So you can see a child is similar with his father. And you can see the dogs. Okay. Dog look like a dog only, no? So what is the reason? What is the reason behind this? The reason is there is a cell, which is a living cell, and cell consists of nucleus. And nucleus consists of chromosomes. So this chromosome is responsible for this heredity purpose. Okay. Cell consists of nucleus. Nucleus consists of chromosome. And chromosomes are responsible for this heredity. Okay. So those chromosomes, they are made up of proteins and nucleic acids, which are the biomolecules we are discussing here. So proteins we have already discussed, and now we are going to discuss the nucleic acid part. So nucleic acid is mainly of, yes, Sandeep, it is because of DNA. Yeah, it is because of nucleic acid, and the types are DNA and RNA. So what is DNA? DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So from the ribonucleic acid, if you remove one oxygen, it becomes deoxyribo, which is called as DNA. So nucleic acids are long chain polymers of nucleotides and are also called as polynucleotides. So what are nucleotides now? The complete hydrolysis of DNA or RNA leads to, or it is because of three. One is because of pentose sugar. The other one is, no. If you remove the oxygen from the RNA, that is RNA is a ribose sugar, right? 5-membered cycle. So 5 carbon you will have. And if you remove one oxygen from, that is you have to remove the oxygen from the second carbon. It becomes deoxy of that ribose sugar, right? So that's why we are calling it as deoxyribonucleic acid, which is DNA. And it consists of pentose sugar, phosphoric acid, and nitrogen containing heterocyclic system. So what are the three components? Consist of pentose sugar, phosphoric acid, and nitrogenous bases. Okay. Now I'll discuss about the sugar. So this was the thing I was explaining to you. This is the sugar molecule, ribose sugar. So this contains 5 carbon, and this is your RNA. This is your RNA. And in the second carbon, you see here, I have removed the oxygen from the second carbon. Now it becomes beta D to deoxy to deoxy. Removal of oxygen from the second carbon. So that's why it is called as 2-deoxyribose, which is DNA. Okay. So coming to the phosphoric acid part, H3PO4 is the phosphoric acid part, and the nitrogenous bases are purines and pyrimidines. So purines consist of adenine and guanine, and pyrimidines composed of cytosine, thymine, and uracil. Okay, so these are the nitrogenous bases. So adenine, guanine, cytosine, thymine, they are present in DNA. So I'll explain this to you in the next slide also. Just listen for a while. And adenine, guanine, cytosine, uracil composed to give RNA. So what is the only component which is changing? Is thymine and uracil. Okay. So the purines consist of both 5-membered as well as 6-membered ring. You can see. You see, we are having one 6-membered ring and one 5-membered ring in adenine as well as in guanine. And all the pyrimidines consist of only one ring, that is 6-membered ring. Okay. Got it? Yes. So now, two different types of sugars I have told you. One is deoxyribose and ribose. And the phosphoric acid part was there, H3PO4. And bases, 5 different bases we have discussed. If a system contains a ribose, that is if a system contains a sugar molecule in addition to phosphoric acid, in addition to nitrogenous bases called adenine, guanine, cytosine, and thymine, if a system composed of all these three, sugar, phosphoric acid, and these are the nitrogenous bases, it is DNA. And DNA is a double-stranded helix. DNA is a double-stranded helix. And here you can see some interaction between the two strands, right? So what are those interactions? They are hydrogen bonding interactions. So always adenine forms the hydrogen bond with thymine, ATGC. So this is the combination. Adenine with thymine, guanine with cytosine. So this is the combination. Okay, ATGC is the code. Only A can form the bond with T. So if this is A, this is T. And if this is G, this is C. So it cannot be of some other type. Yeah, glycosidic phosphoester linkages, I will explain you. I will explain, Sandeep. I will explain. So this is about your DNA. And coming to RNA part, you will have sugar and phosphoric acid, adenine, guanine, cytosine with uracil will be there. So all these composed of RNA, it is a single-stranded helix, you can see. And coming to the next part, which is nucleoside and nucleotide. So the nucleoside attachment of a base to 1' position of sugar. You can see here, the first carbon of your sugar molecule is attached with the base. And if a system has only sugar and base, it is called as nucleoside. If a system has sugar, base as well as phosphate group attached to it, to the 5' end of your sugar, it is called as nucleotide. Okay. So sugar plus base forms nucleoside. Sugar plus base plus phosphate group is your nucleotide. Got it? Shall I go for the next slide? Yes. Now I'll explain you the bonding in nucleotide, which you were asking. Okay. So this is a nucleotide which contains sugar, base, and phosphate group, and it is now going to treat it with another nucleotide. Now two nucleotides are going to interact with each other, and a new bond is going to form between the phosphate group of one molecule and the 3' end of the other molecule. The removal of water occurs here and the oxygen linkage is formed. between the two systems. So H2O is removed and you are getting an oxygen linkage between phosphate and 3' end of the other nucleotide carbon. Okay. So this linkage, the 5' end and that is the 5' end only contains this phosphate group, no? That's why it is called as 5' end of the chain, and 3' end of the chain forms a linkage called as phosphodiester linkage. It's called as phosphodiester linkage. Sandeep, is this clear now? Phosphodiester linkage is clear to you, right? It is existing in nucleotide. Clear? Shall I go with the biological functions? Okay. So reservoir of the genetic information, one of the main biological function of nucleic acid, and the identity maintaining of different species, and DNA molecule is capable of self-duplication, which is called as replication, right? And DNA contains the message for the synthesis of protein, and RNA synthesis the protein. RNA synthesis your protein. So protein synthesis is done by RNA. So these are the biological functions of nucleic acids. Okay. So there are some differences between DNA and RNA shown to you on the screen. You can take the screenshot of it. Just take the screenshot of it. The last biomolecule is hormones. Shall I discuss the hormones now or in the next class? What do you say, Sandeep? I think, yeah, I'll discuss the hormones in the next class. And I'll start with the new chapter in the next class, okay? I'll start with the new chapter called D and F block elements to you in the next class, okay? So thank you so much, Sandeep. I think you don't have any doubt here so far in the class. Meet you in the next class. I'll meet you tomorrow, okay? Thank you so much.